Hello, welcome to the first video. This unit is covering how do cells convert food energy into ATP. And we're going to be talking about aerobic respiration, anaerobic respiration, and at the very end, a very brief description of how plants convert energy and how they actually produce their own food in the form of glucose, and that's photosynthesis. So to give you a little bit of introduction, all cells use ATP for energy. Now, what are some examples of, different kinds of movements and things like that? And all cells require ATP. You can see in this video, you see the guy sprinting, very powerful movements. You can see how fast they're running. That is using a pathway called anaerobic respiration, and in some ways it's actually called the creatine phosphate pathway, but you can see how quickly they're running. Their movements are being powered by a process that does not involve oxygen. You can see in this particular example, see how much slower the running is. This is a distance of it, it's a 3,200 meter dash, and they're using a pathway called aerobic respiration. It's for lower intensity exercise, daily living activities, even though they're still running fairly quickly for it to be a long distance race, they're using a different pathway. Now, in this example, this is a neutrophil, which is a white blood cell. And what it's doing, somebody got really bored in a lab and they added some Staphylococcus bacteria, and they videoed this neutrophil chasing this Staphylococcus around. And what a neutrophil is, it's basically a Pac-Man cell. It goes around eating other cells. It's part of our immune system. And you can see when it's moving that, it requires energy. So it is also breaking down energy in a process, and is powering the cell to be able to go around and catch that staph and then break it down. And even the Staphylococcus bacteria, they also have the same kind of pathways to convert energy. So let's go ahead and get started. So we're back on our outline. Now, here's the first thing. Big picture. This is adenosine triphosphate. It's an adenosine and these three phosphates all attached. And what happens is there are enzymes, and these things we're talking about today, they're all powered by enzymes. There's an enzyme that comes up here and breaks this little bond. These are all held together by chemical bonds, the stuff we talked about last week. And the enzyme is called ATPase. It's ATPase. Any word that has ASE on the end as a suffix, the word previous to it is an enzyme. Big word. There's an enzyme in our neuromuscular junctions, the myoneural parts where the muscles connect to nerves. There's an enzyme there called acetylcholinesterase, ASE, that breaks down the neurotransmitter acetylcholine. So, enzymes power all this stuff. We won't talk about that. But when that bond is broken, that's what cells do, there's an energy release, and you end up with this thing called adenosine diphosphate, ADP, and you end up with that little phosphate that was attached here. But when that bond is broken, you get this big release of energy, and cells capture that energy to power their functions. Another good diagram, so take a look at this when you get a chance. We talked about these categories last week. Here's food, polysaccharide, fats, proteins. Polysaccharides are broken down to sugars, which then can be used in glycolysis, which all of these can. And these are three examples of how a cell will convert food energy into ATP. This is the Krebs cycle. This is called the electron transport chain. All of this takes place inside a little organelle called a mitochondria inside of a cell. This takes place in an area called the cytoplasm, which is outside of the little organelles that make up cells. Now, I'm going to jump over here real quickly. Sorry, I clicked on the wrong. There we go. So really quick, just background. This is a cell. We're actually going to cover this during our semester. There's a nucleus, there's a nucleolus. All this area out here is the cytoplasm. In fact, these are mitochondria. So this is what we're talking about today. When we talk about the Krebs cycle, the electron transport chain, all that takes place inside these little organelles. So that's an overall cell. The other thing I wanted to touch on real quickly is this. I'm going to make this big screen. Now, this section we're covering in biology is a hugely detailed section. We're going to do it very general. But I wanted to give you this idea. This is how a hormone interacts with a cell that can't cross a membrane. But see all this in here? That's what we're going to be covering in this section. When cells convert energy, you have all these reactions going on inside of a cell. Some of them happen inside of little organelles, whether that's a plant cell or an animal cell or eukaryotic cell. And then some of it takes place out here in what is called the cytoplasm. And that's glycolysis and like anaerobic respiration, the other part we're going to talk about. Okay, so just imagine this. When we're going through all these pathways, okay, this goes through here and this is what's produced. It looks something like this. You have these big chemical reactions and then down here that are going on. So let's go back here. Now, cellular respiration or aerobic respiration uses oxygen to power all of this, okay? It takes the oxygen that, if you're talking about human beings, that we breathe, and the food that we eat, and it goes through this little pathway to convert ATP. And it's the most efficient way of doing that. There is a different pathway called the anaerobic pathway or anaerobic respiration that does not involve oxygen. It doesn't mean you're not breathing. It just means that oxygen is not involved with the part where you're converting energy. So, first things first, this is a mitochondria, like I showed you just in that previous diagram. And that's where the aerobic respiration part of this is going to take place. So like it says here, elongated organelles inside cells that are responsible for converting energy into ATP using oxygen. Oxygen is the last electron acceptor. Excuse me, I'm sorry. Another cell diagram. Here's there's some more mitochondria. We'll talk about all these other structures later, but just a little smaller version of the one I just showed you. Now this whole thing is powered by enzymes, and you have to know what an enzyme is, how do they interact, and that's really about it. Very simple explanation. I usually draw these on the board. I'm not going to do that. I have the ability to try to draw with a mouse on my computer. I think it looks worse, so I'm going to use a diagram. So once an organism has acquired nutrients, these nutrients can be utilized for cell respiration. And you have to have an enzyme to power this whole pathway. So this whole thing up here, and I'll go back, this is all powered by enzymes. Enzymes are breaking food down into these categories that are smaller, and then enzymes are powering this whole process. All right, so you have to know a little bit about what is their structure and how they work. Very simple diagram. Here's an enzyme, and then enzymes are biochemicals. You can't even see them. If you wanted to see them under a microscope, you can't. And if you were to Google an enzyme, you're going to get this swirly looking structure. They're proteins. Remember the tertiary and quaternary protein levels where they're all twisted around? That's what an enzyme would look like. But they have a very specific shape. And here's the first part of this. So, well, like I said, once they've acquired nutrients. Now the substrate is very specific, and it's what the enzyme is acting upon. You see the shape here, and okay, a binding site or active site on an enzyme is where the substrate binds, and there's a very high affinity here. There's an attraction. It's like a lock and key example. Every enzyme has a very specific shape, and it's going to bind to a very specific substrate. I have an example for that. So here's a substrate, and you see there now it's bound together. This is actually now called the enzyme substrate complex. Enzymes are not really altered during the experiment. Most of the time the changes occur to the substrate or maybe to a part of the cell. And I'd mentioned this. Enzymes usually end with the word ASC. Not all of them do. But yeah, the term means, ASC on the end of a word usually refers to some kind of enzyme. So I'm going to give you this diagram. This is showing how if you eat something that it's either a sugar or a polysaccharide. Remember last week, this is called sucrose. And sucrose is a substrate. See it over here? Remember it's a disaccharide, 2 sugars. Sucrase, which is this, and see the active site. Sucrase is an enzyme that breaks down sucrose. And it says right here, sucrose consists of glucose and fructose bonded together. Our bodies, and including bacteria, they have different parts, but our bodies can't absorb sucrose. It's too big. It would just, if you didn't have this enzyme, it would sit in the small intestine, and then the microbes there would break it down, and then it would cause pain and gas and all kinds of things. So here's how this works. So this represents sucrase, ASE, and I thought it doesn't say that in our little diagram here, but you see sucrose, see the shape, see the enzyme, and then boom, here's the binding, the lock and key thing. You get a little byproduct of water. Every chemical reaction in the body has water as a byproduct. Or in this case, this is actually incorporating water into splitting these two apart. It's actually called hydrolysis. Hydro means water, lysis means to dissolve. But don't have to know that. But see where it's bound together, and this takes a little bit of this water molecule and it splits it into glucose and fructose. Now that's small enough to be absorbed. And see the enzyme breaks off and it's not affected. And what you have here are called products. And we'll go through all the products of every part of respiration. So anyway, that's how that works. All right, so let's go back on our outline. At any point, if an enzyme the shape gets changed, the term is it's denatured. That's when you alter the shape of a protein. And when you do that, it is no longer functional. I actually have a PowerPoint that has a great example for that here. Functional protein, denatured protein. See how it's all different shapes? And then if you denature it, of course, you're not going to get this because now this active site area has been altered and it's not going to bind together. You're going to do an experiment that's going to show that, a lab experiment. There are factors that cause this substrate enzyme binding to be altered, changed, or destroyed altogether, or even stopped. Freezing actually stops enzyme activity. But if you have something that's too hot, like when you cook an egg and it all sticks together, that's all the proteins coagulating. And so, yeah, at that point, they're destroyed. They're not going to work. So one more example, enzyme active site. Here's a situation where this is called anabolic, where you're taking 2 smaller particles or 2 smaller substances and you're making a larger one. Going this way is actually called catabolic, and it's on those terms at the end of this list. But when you have a large molecule and you split it into 2 smaller ones, that's catabolic. Now, last part of this video, and I'm going to stop and start with the next one, but under what's called enzyme activity, there are things that affect that, but this is a great example. This is energy. It says progress of reaction. You see this big arc up here. All chemical reactions, including this one, this little binding to here, that requires, it's called activation energy. That's the energy required to perform the reaction. And you see this big arc? It says right here, activation energy without an enzyme. See how much more energy is required? In fact, a lot of times these reactions, the activation energy is so high that they would not occur unless there was an enzyme involved. This would not split into those two molecules, most likely by itself. This is where the enzyme comes in to help that. And enzymes, see this? And this is so funny because all we hear now today, I'm gonna date this video, but we're talking about flattening the curve of our pandemic. You see this flattened curve? That's what enzymes do to the activation energy. And then you end up with products, which would be, if you're going this direction, these are the products. All enzyme reactions are reversible. So you can take two smaller ones, build a larger one. So that's why the arrows are going both ways. But that's what enzymes do. They catalyze chemical reactions. They lower the activation energy, allow them to occur. Without an enzyme, most reactions would not occur. Now, you're going to do an experiment for this in lab. What are some factors that affect this right here? Okay, what affects this? pH, remember the scale we went over last week? It goes 0 to 14. Every organism has an optimal pH, and it does vary depending on what you're talking about. If you remember human blood on that scale we were looking at, one of them, human blood is about 7.35 to 7.45, so it's slightly basic. If the pH in blood starts getting too acidic, okay, the number gets smaller, or too basic, the number gets bigger, it will start having effects on enzyme activity. And the first thing that happens in acidity or alkalinity is proteins start doing this. They'll start denaturing, and then the enzyme activity will be altered. In most cases, it's going to slow down. So basically, pH, either more alkalinity or more acidity. In other words, a decrease in pH means it's getting more acidic. The number is getting smaller. An increase in pH number means it's getting more basic. But both of those cause enzyme activity to decrease. Okay, it affects the bonding between the molecules because the shape of the enzyme starts changing. Temperature. An increase in temperature usually speeds up, well, it does. It speeds up enzyme activity. But there'd be a point where the higher temperatures would start doing this. You start getting denaturing, and then it would go down. But in a microbiology lab, you have Petri plates with agar, and you swipe things on these agar plates, and you put them in an incubator. The incubator is set at our body temp, 97 degrees Fahrenheit, or 98 degrees Fahrenheit, 37 degrees Celsius. They will grow faster at that temperature than they will at room temperature, which is 20 degrees Celsius. However, if you got too hot, yes, at some point, the enzymes would denature and it would kill the cells. Decrease in temperature, they slow down enzyme activity. So this little procedure, this reaction right here would go slower in colder temperatures. Freezing stops it altogether, 100%. And freezing would, yes, kill some cells. Human beings, right, we would get hypothermia at some point. Our metabolic rate would drop to a point where we couldn't sustain ourselves. Substrate concentration. Now this is kind of a obvious one. These represent enzymes. This is low substrate, and you see here is kind of moderate between the two of them, and then here's high substrate. So the amount of available substrate affects enzyme binding. Of course, here you're going to have less binding of enzymes to substrate. Here, in extremely high substrate, you can see they're all bound together. So Increasing the availability increases enzyme activity. And the last one of these in this video, we're going to go to the next video, is there's a thing called competitive inhibitors if you're talking about enzymes. And what these are, when a substance competes or prevents normal binding of the substrate to the active site, that is called a competitive inhibitor. See the blue ball of our diagram earlier? See, now it can't bind to the active site because now it's got a competitive inhibitor sitting there. That's how poisons work. That's how cyanide works. Cyanide kills cells, animals, humans. Cyanide blocks an enzyme called cytochrome C oxidase. And I'll show you where it happens here in a minute. And it binds here. There's no more normal binding during the pathway of converting energy, so the person can't convert energy. The Botox toxin is a competitive inhibitor for acetylcholine, which is a neurotransmitter. Yeah, a lot of poisons work that way. So that's what a competitive inhibitor is. It prevents binding of the normal enzyme to substrate complex. Last example. You're not supposed to sit in the garage with the door down and your car running. Cars produce carbon monoxide, M, not dioxide. And carbon monoxide binds to hemoglobin in the same area that oxygen does. In fact, it has a stronger attraction to it. And so essentially a person would pass out. Their blood would not carry as much oxygen and they would asphyxiate. So, okay. The next video will start right here with cell respiration.